Hi, this is podcast number 11. And in podcast 11, 12, 13, we're going to talk about different elements of the cytoskeleton. As I mentioned, the cytoskeleton is very dense inside cells. The cell you want to think of as a soggy Nerf ball, not like a bag of vegetable soup. There's tons of protein in there. A lot of that protein is filamentous. This is a scanning electron micrograph of the cytoskeleton with its different fibrils that are given false color. It's not like they have color inside your cell. This is just to make things look easier. The thickest ones here are microtubules. The thinnest ones are actin filaments. And then the intermediate ones in blue here are intermediate filaments. And you can distinguish among these three types of filaments by what kind of proteins they're made out of and also their size. So today we're going to talk about microtubules. Microtubules are made out of the protein tubulin, either alpha or beta tubulin. These are two types of tubulin that have different amino acid sequence, but they have very similar three-dimensional shape. And they have similar characteristics in that they both bind GTP, and they form these large diameter filaments, 25 nanometers across, and the filament is hollow in the middle. In the other podcast, we'll talk about microfilaments and intermediate filaments. Microfilaments are the thinnest filaments, not hollow in the middle, and they're made up of actin, protein, and intermediate filaments are intermediate. They're in between microfilaments and microtubules, and they are made up of a whole bunch of different kinds of protein. Okay, so one characteristic of prokaryotes versus eukaryotes is prokaryotes don't make tubulin, but they do make a molecule that looks very much like tubulin. It just doesn't share much sequence, but its three-dimensional shape is very much like tubulin, and it acts like tubulin in that it binds GTP, and it forms these filaments. In prokaryotes, it's called FTSZ, and in eukaryotes, you have tubulin. So that's another difference between prokaryotes and eukaryotes. So continuing on with eukaryotic tubulin for the rest of this podcast, a tubulin will polymerize into a string of alpha and beta subunits bound next to each other in these doublets, alpha, then beta, then alpha, then beta, with the help of GTP and magnesium. This one chain, alpha, beta, alpha, beta, alpha, beta, on up, is called a protofilament. And if you put 13 of these next to each other, you get a microtubule. So 13 is the most common size, but there can be more, can be less. If you have one single microtubule, those are called singlets, but you can also have doublets where you have another tube attached to the first tube, and even triplets where you have three tubes attached to each other. And this second tube incorporates some of the protofilaments of the first tube, so it's not quite 13. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 new protofilaments added to the original 13, and then another 11 to get to a triplet. All right, one thing that can be confusing about microtubules is how they grow and how they shrink. So they're on the individual alpha and beta tubulin monomer units. There's a polarity. There's an end that is on one end that is different from on the other end. And on one end, there'll be a GTP binding site That is not on the other end. That binding site is the end that tends to add on new doublets, alpha and beta subunits, this doublet here. But and if that, because that end tends to add on new doublets, it tends to be growing, and that's called the plus end. And the other end, which tends to lose doublets, is called the minus end. However, saying that, both the plus end and the minus end can lose or gain at any one time. It's just on average, the minus end is shrinking and the plus end is growing. Okay, so this depends on many things inside the cell. It depends on the concentration of free doublets. It depends on the concentration of GTP. On the plus end, it takes a lower concentration of free doublets to do a net addition of units to this end than it does on the minus end. So if the concentration of free doublets in the cell is fixed at a given amount, then the plus end is going to add things faster than the minus end just because of the affinity of the plus end for these doublets versus on the minus end. But if that amount of free tubulin changes, then the growing and shrinking of the microtubules can change too. So initially in the cell, when it's going to form a microtubule, you have individual doublets that form these small chains of small amounts of doublets coming together. Those are called oligomers, and that takes a long time, so there's a relatively flat curve in terms of how many microtubules are being formed. Then when you start to get a protofilament forming, the microtubules tend to grow rapidly, and so you get this steep rise in the growth of microtubules during this period. And then as you run out of extra tubulin doublets, the microtubule will start to shrink. And the whole time the microtubule is adding and subtracting from both ends. It's just adding faster to this end and subtracting slower from that end. If you look at what this looks like in a micrograph, you have a basal body, which is a microtubule organizing center. I'll talk about that in a second. That's essentially where the microtubule starts growing from. And on the plus end, it is growing faster than it is on the minus end. So it's not that the minus end can't grow, it's that it grows much less fast than the plus end, and that's why this is called the plus end. There is a phenomenon that can happen where you are adding things to the plus end, and at the same time you're subtracting things from the minus end, such that if we followed individual microtubule doublets, tubulin dimer doublets, and follow the fate of those as this microtubule matures, you'd see that they start off on the plus end and then they end up being on the minus end just because you're adding things to this end, the plus end, and you're subtracting things from this end. So as a result, eventually they will end up on this end. And this is called treadmilling. There are common drugs that are used in cell culture, actually also in medicine, that affect microtubules. There is a drug that comes from a plant, the autumn crocus, called colchicine, and it inhibits microtubules from forming. There's another one called vinblastine that also inhibits the growth of microtubules, and then there's Taxol. You might have heard about that. And then what this does is it stabilizes microtubules, prevents them from destabilizing, and therefore doesn't allow microtubules to change. So a lot of cell functions are associated with microtubules growing or shrinking, and so if they're stabilized, that cell's function stops. So you may have heard of this one before because that's a chemotherapy drug. And microtubules are used to separate chromosomes during mitosis. And if you stabilize those chromosomes, then or stabilize those microtubules, then you arrest mitosis. And during um, cancer, you have uncontrolled cell mitosis. So uncontrolled cell growth, you can stop that with Taxol. However, there are cells that naturally are replaced all the time, and they're growing all the time. And if a person is taking Taxol, they will affect those cells also. So some of the common side effects that you know about with chemotherapy, such as hair loss and nausea, those come from cells that are constantly dividing, such as in the roots of your scalp and in the lining of your gut. And if you prevent them from constantly being replaced, then you get sick to your stomach or your hair falls out. Okay. But we can use these in cell culture to try to figure out how these microtubules work by chemically making them stable or chemically making them fall apart. The growing end, the plus end of a microtubule will have GTP bound to it. And then as that growing end gets old and starts to move toward the minus end because of treadmilling, that GTP can be hydrolyzed to GDP. The rate at which the GDP is formed from GTP versus the rate at which new GTP is added depends on how fast the microtubule is going to grow or not grow. So if there's a ton of GTP tubulin around to add to this end, then the microtubule will grow faster than it'll shrink. If the rate at which the GTP gets hydrolyzed to GDP is faster, then the microtubule will tend to shrink. At the point where you exhaust the GTP cap, that there's no tubulin subunits with GTP bound on the plus end, you can get what's called catastrophe, where the whole microtubule falls apart at once. And these complicated, is it growing, is it shrinking, all depends on multiple equilibria. So it depends on how much free tubulin is available, how much of that free tubulin has GTP bound to it, what is the rate at which GTP is being hydrolyzed to GDP. And so if you follow the length of an individual microtubule on the plus end, for example, you can see that it's getting bigger and then it gets smaller and then bigger, smaller, smaller. And it can change dynamically as you watch it under the scope. On the minus end, not as much. There's more action on the plus end than on the minus end. Together, this is called dynamic instability, that the size of the microtubule changes all the time. Okay, here is a H5P question. Okay, so we mentioned that that there is a thing called a microtubule organizing center, or a place where microtubules start. In animal cells, this area of the cell is called a centrosome. So these terms are very confusing, and people get them confused all the time. I'm going to try to make it crystal clear here. Okay, so there is a structure in animal cells that has this structure, which is a ring of triplet microtubules. Very unusual looking structure, and has one, and they're short, one facing in one direction and another one facing at a right angle to it. That's called a centriole, a pair of centrioles. And these duplicate and migrate to opposite poles when a cell is getting ready for mitosis. Okay, only animal cells have those centrioles. The area around where the centriole is called the centrosome. So like the home base in the cell for a centriole is a centrosome. It's just the area around the centriole. So a lot of this comes from what we can see in an electron microscope. We can see this structure really well because these stain really well, but we can't see a structure around this area because it doesn't stain that well. And we call that area the centrosome. This centrosome is a type of microtubule organizing center. So microtubules grow out of this centrosome because it is a microtubule organizing center. So plant cells don't have centrioles, don't have a centrosome, but they have a microtubule organizing center. They have microtubules. They extend their microtubules during mitosis, just like you would see in an animal cell, except there's just not this structure that we can see and not a centrosome. It's just not called a centrosome in plants. But there is an area from which microtubules grow called the microtubule organizing center. So microtubule organizing center is a very general term, meaning microtubules grow from this part of the cell. There are different types of microtubule organizing centers. One type in animal cells is called a centrosome, which contains within it a structure called a centriole, which is made up of two of these things, okay? Two of these microtubule structures, okay? There are other types of microtubule organizing centers, and we'll talk about those. So again, I want to stress that the centriole is part of the centrosome. The centrosome is the area around the centriole. And microtubules grow from that area, and that area is a type of microtubule organizing center. Plant cells don't have a centriole or a centrosome. They have a different type of microtubule organizing center. And microtubule organizing center is a general term that applies to anything from which microtubules grow in a certain direction. So in an animal cell, when you slice through the centriole, you'd see this sort of picture. So this one is coming out of the slide at you. This one is going at a right angle away from you. They're at right angles to each other. So those are the centrioles, the two microtubule structures. The area around it is called, in this slide, pericentriolar material, but this would be the centrosome, and then microtubules grow out of the centrosome as a type of microtubule organizing center. Here is a nicer picture of a centriolar structure. So this would be one centriole, these triplet structures arranged in this very beautiful pattern around the central axis. Okay, from a centrosome, the negative end of the microtubules will start, and the positive end of microtubules will always be facing away from the centrosome. There is another kind of tubulin, gamma tubulin, that is important for initiating these microtubules at the minus end. And that gamma tubulin is only found in centrosomes. So up here in the microtubule, these are alpha and beta subunits of tubulin. Gamma tubulin is a different type of tubulin, and you only found that in centrosomes, and that's where centrosomes get started on their minus end. So in all microtubule organizing centers, whether it's a centrosome or another kind, the negative end will be at the microtubule organizing center, and the positive end will be growing away from the microtubule organizing center. Okay, here's an example of that. Here's a neuron, nerve cell, with its nucleus. It has a centriole, paracentrioles, okay? And the area around that is the centrosome. That's a microtubule organizing center. The microtubules are going to grow out from that and the plus N will be far away from them. The minus N will be at the centrosome. And the centrosome is a microtubule organizing center. There's one on the side that forms an axon that has all the microtubules are in the same orientation, that all the minus ends are toward the cell body, all the plus ends are away from the cell body. And dendrites, which are a different process coming out of neurons, they're not as well organized as that because there's no MTOC over here. This orientation of the microtubules, we're going to see soon, is super important to the function of these axons. Remember, these axons can be really long as your leg, and that all the microtubules are in the same orientation is really important for transporting things along that axon. Okay. Once again, the microtubule organizing center is the most generic name for a place in the cell that generates microtubules. And at the microtubule organizing center, you have the minus N, and far away from that, you have the plus N. If you don't have microtubule organizing center, the plus N and minus N can be all over the place. So if you think of the microtubule organizing center as the root of a tree that anchors the microtubule with its minus N, but it's not the only place in the cell where you can grow microtubules. But just if you have an MTOC, they will all be in the same orientation, which can be handy. Here's another example of a place where it can be handy. So in ciliated cells, like in the brush border of your gut, sorry, not in your gut, but in because that's a different type of cell, but in like the cells lining your throat. Okay, those are ciliated cells, and they are beating. They have a basal body at the base of these finger-like projections that are cilia that are coming off of the cell. All the microtubules have the same orientation in those cilia. The minus N at the basal body, the plus N at the tip of the cilium. And that's because of these microtubule organizing centers. In this case, they aren't called centrosomes, they're called basal bodies, but it's just another type of microtubule organizing center. Okay, and again, the minus N is always at the microtubule organizing center and the plus N far away. So why is that important? Because they're all in the same orientation. We will see later, motor proteins are going to work on these microtubules to cause them to bend and cause the cilia to beat in a coordinated fashion. So, like I said before, you don't need a microtubule organizing center in order to have microtubules. You can have microtubules without one. In red blood cells, they're shaped sort of like a lozenge, like a throat lozenge, and they get that shape from having a band of microtubules that goes around the outside. It's a less organized band because they're not all in the same orientation because there's no MTOC, but they're there nonetheless. The orientation of microtubules during mitosis is important. So there's in mitosis, the centrosome starts growing microtubules, the minus N at the centrosome, plus N forming away from that as the cell forms the spindle. And during metaphase, you'll have these duplicated centrosomes forming the poles of what will be the two new cells. Both of them have their negative ends, and then microtubules growing out from them, which the kinetochore microtubules will attach to the chromosomes and start to pull those apart. So we'll learn more about that later. I just want you to know that, again, orientation of the microtubules, plus N and minus N, is important for this cell function. There are other proteins associated with microtubules besides just the alpha and beta and gamma tubulins. These are called, in general, MAPs, which stands for microtubule associated protein. Tau is one of those kinds of MAPs. EB1 is another kind of MAP. TIPs are another kind of MAP. Catastrophins are other kinds of MAPs. So these are proteins that bind to microtubules and help in their function. Taus help microtubules space apart from one another, which is also really important in neuron function. And taus have been implicated in Alzheimer's disease and Parkinson's disease because it forms these giant oligomerizations of phosphorylated versions of this protein called tau tangles that are characteristic of people who suffer from these diseases in their central nervous system. Other microtubule-associated proteins can be associated with stabilizing microtubules, preventing them from falling apart or causing them to fall apart when a cell is being remodeled. So like when a cell is going to move, for example, it'll depolymerize their microtubules in order to allow it to move. Okay, here's another one for you.